Okay, so we are on part 4. We are moving along. All right, we will start. And let's just a brief recap before we get going on the hormones and the nutrition of how glands are doing their business and how hot dogs and whatever other nutritional uh, factors are working. Just a quick recap at what we're looking at. Right? So we have, let's just say it's muscle, and I mean, we have cell surface receptors all over the place. It doesn't matter what cell it is, you're going to have some cell surface receptor. But let's say we have a sarcolemma and a skeletal muscle fiber, and inside of it you have PKB. Inside of it you have PKB. Downstream from PKB is all this mTOR signaling, and upstream from mTOR signaling are all these activators. And at the sort of very top, we have the cell surface receptors. So if we have an insulin receptor, it's a bunch of PI3K signaling. And remember, PI3K is going to convert PIP2 to PIP3, and then PIP3 is how we recruit PDK. So PIP3 is how we're going to get PKB phosphorylated. PDK is going to do that activation of PKB, but PIP3 is going to recruit it, and PI3K is what's going to do that. And P10, phosphatase and tensin homolog, P10, I'm not going to ask about this one, but this is just doing the opposite of PI3K. You're converting PIP3 back to PIP2. It's a phosphatase as opposed to a kinase. Remember, a kinase, you're attaching phosphates to things. These phosphatases, you're taking them off. And so the initial beginning of all this, and then at the end, we have mTOR phosphorylating. mTOR itself is a kinase, and it's going to phosphorylate its downstream targets. And so you see P70 S6K right there, ribosomal protein S6 kinase, P70 S6K. And this diagram doesn't have 4EBP1 on it, but that's another one, right? That's an inhibitory effect. When mTOR phosphorylates that one, eukaryotic translation initiation factor. It's a binding protein. When mTOR phosphorylates 4EBP1, um, it is halting its inhibition of translation. So mTOR phosphorylates P70S6K, which has a ton of names, remember, and that one is going to initiate, well, you see ribosomal protein S6, it's going to phosphorylate that because this too is a kinase. So it's going to phosphorylate this thing. And now we're really getting started on translation. Translation, linking together amino acids. Translation, hypertrophy, protein accretion, creating your proteins. And so the overall map, I referred to this canonical signaling, this PI3K to PKB to tuberin, Rheb, mTOR, P70SSK. This as I5, right? You're going from the cell surface down to protein translation, down to hypertrophy, but you're doing other stuff in the process. This is what's inhibiting GLUT4 translocation, and so PKB is phosphorylating that to withdraw its inhibition, and FOXO, fork-head boxo. We talked a little bit about that. We'll talk a little bit more about it on Friday. Glycogen synthase kinase 3. That's the thing that's halting glycogen synthase to create your glycogen to start amassing more glycogen. So PKB stops the thing that's stopping the thing. There's so many double negatives in regulation, and so I'm turning off the thing that turns off the thing, and so thus I'm turning it on. And as we get into the nutrition, the amino acid part, we really know carbohydrate metabolism, some of it, a lot of it, because you get insulin, right? You get a bunch of carbohydrates into your bloodstream, you get blood glucose, and that's the trigger for your pancreas, for those beta cells in your pancreas to spit out some insulin. And insulin is going to bind to its receptor, and then you get PI3K signaling. So you know how that works. But we're going to do the protein side today. As soon as we're done with sort of the rest of hormones, we'll do the rest of hormones. We've been talking about insulin, so there'll be one slide on insulin because you know about it. We've been talking about this sort of all block. So we'll have one slide. on that. But we'll talk about the other stuff, testosterone and estrogen, and really mostly those two. We're gonna talk about, and and then we're going to get into amino acids. You eat a bunch of proteins, and what does that do? So this is where we left off last time. We did chemicals and we did mechanical loading. We did this tension, the mechanical forces applied, all this integrin signaling, mechanotransduction, all of that stuff, and the chemical sort of immune response of things. We talked about interleukins 2 and 15. 6 is weird. 6 is like hypertrophy and atrophy. So I don't like to talk about that one. I like stuff that has a clearer nature. Although I did mention reactive oxygen species just because they play in so many areas. I'll bring them up again today, but reactive oxygen species are a little bit finicky, and I don't expect you to know really anything about them for the exam, but to be familiar with their presence. In every TV show or movie or book, there's going to be minor characters, and you don't actually even have to know their names. You can just go, though the chef, you don't have to know their backstory, you don't even have to have a name. And so think of reactive oxygen species at that level, but I'm going to keep talking about them at the very end of the semester when we're talking about mechanisms of fatigue. They'll make a reappearance. So just it's important, but they're a little bit finicky. But today is just endocrine and nutrition, and I'll do my best or something close to my best, maybe not my very best, to get us through these topics in a reasonable amount of hours and minutes and seconds. So endocrine system, right? We're going to begin with the endocrine system, finish with nutrition. There'll be a little bit of overlap in these things, some sort of a little bit of crosstalk, if you want to call it. But hormones work. The endocrine system, classic endocrine system, not just immune, like, interferon gamma and IL-2 and 15 and tumor necrosis factor and prostaglandins, the stuff that's sort of classic immune tissue disruption and the healing cascades, not just chemical things, chemokines, but glandular responses of what we think of endocrine. We're secreting hormones into the blood, sometimes autocrine or paracrine, but big endocrine responses and what's going to happen here. And when you see the Ronnie Colemans of the world, the endocrine system works. I mean, this really works. I mean, this is like a world champion in natural bodybuilding, right? Put him up against Ronnie Coleman, and it looks like a different species, right? It looks like we have, okay, over here we have a prehistoric upright bipedal, well-oiled, tanned, lean hominid, and then on this side we have Ronnie Coleman. But hormones work, and we talked about this in our steroid arguments of that sort of silly but well repeated argument that, well, steroids, androgens, exogenous androgenic anabolic steroids don't do anything, so why would you do them anyway? They obviously work. They do a lot. And the way that they work, the hypothalamic pituitary gonadal axis. Get to those gonads, right? Testosterone, get those testes to produce their sauce. And so this is the hypothalamic pituitary gonadal axis. And luteinizing hormone is the cutoff man for the play at the plate to get there. And testosterone is going to bind to its androgen receptors. And primarily this is cytoplasmic, right? This is get inside of the cell. This isn't cell surface, although I'm going to talk about some exceptions here. But the primary effect of testosterone, it is a steroid hormone. It's cholesterol made, and it's going to cross that lipid, that phospholipid bilayer, that membrane, get into the cytoplasm and bind to an androgen receptor there, and it will change its shape a little bit, take a conformational change, right? So this receptor and testosterone complex is going to do a little bit of a shape change, is going to like transformer itself a little bit and bind to the hormone response element, right? But it's an androgen response element, and it's going to regulate transcription. But I don't really care that much about that piece of testosterone. Yes, it works. Sure, it's really important. But as we talk more and more about fitness, the way I talked about it last lecture, the lecture before, or whatever, how we have a metabolic fitness, a protein turnover, and how we're regulating the size of our cells, right? How we're regulating how much hypertrophy and atrophy we have. Is synthesis outpacing degradation, or is the inverse true? This is mostly cell signaling that's responsible for this interaction of the human form and its environment. And there are secondary effects of testosterone that are non-genomic, right? These are different. This isn't like, oh, let's bind to a little patch of DNA and affect transcription. There are non-genomic effects of testosterone and a bunch of second messenger cascades. And part of this, you're going to see cell surface receptors and so androgen receptor, but this is the cell membrane, right? And so this is G protein androgen receptors. And so let's get phospholipase C activated. You don't need to know this cascade, but I'm going to walk you through it a little bit. Inositol triphosphate levels down here, which are going to bind. This is a sarcoplasmic reticulum. What does to a receptor on the sarcoplasmic reticulum? What's stored in the sarcoplasmic reticula, plural, calcium, right? So this is a little cascade, a little signaling cascade to release calcium inside of the cell. And that calcium seems to activate MAPK signaling, mitogen activated protein kinase signaling, which has a convergence with mTOR at the end. They are doing the same stuff, hypertrophic stuff at the end, but also there's crosstalk in between. The MAPK signaling cascade is crosstalking. It's communicating with mTOR at raptor and at tuberin, the tuberous sclerosis complex, but specifically TSC2, which is tuberin. And so with these cell membrane receptors, testosterone seems to elicit a calcium release from inside of the cell, and it matters, right? This cell signaling cascade effect of testosterone matters, and a lot of the effect of testosterone and regulation of muscle metabolism seems to not just be that calcium release, but also insulin-like growth factor. Testosterone has there's a synergistic response between testosterone and insulin-like growth factor, IGF, and a potentiation of the signaling, an increase in the amount of IGF that you're getting, and that corresponds to muscle growth. And you'll see, I mean, this is like, animal diaphragms, but you'll see this in skeletal muscle. You can see a similar effect of the higher threshold motor nerves you're experiencing, or muscle fibers, you're experiencing more hypertrophic signaling and more and more IGF in those. So when you see a control group, right, you're not receiving nandrolone, you're not receiving this exogenous steroid, right? This isn't an endogenous thing. This is, let's give people exogenous steroids, give rodents exogenous steroids, and you see more muscle IGF-1, more of it. And the consequence is where you're seeing more of it is in those higher threshold fibers. You remember when we went through the fiber types, you get into those higher threshold fibers, and you're seeing both more IGF-1 and more hypertrophy. So a lot of the testosterone effect is not just the release of calcium, but it's also IGF, insulin-like growth factor. And we've known this for quite a while. In 1993, testosterone administration increases IGF levels in normal men in the 90s, right? We've known this stuff for quite a while, but you start fast forwarding the world. Here's 2013, and spin that world a few more times. Wrap it around the sun a few more times, and we start looking at testosterone's effect on mTOR. And in part, we see things like the calcium release. Right, we're going to see that intracellular calcium release from the sarcoplasmic reticula and the MAPK signaling, but also we're getting that insulin-like growth factor, PI3K, PKB, mTOR signaling, and then the classic testosterone stuff, androgen receptors, these cytosolic receptors, and nucleic effects. So testosterone is multifaceted. Testosterone works in multiple ways, and the ways that I care about for this class are on this side, and especially this one. But MAPK and PI3K, PKB, those two roads, testosterone has a big effect on signaling through those two roads. Another thing that testosterone is doing is inhibiting atrophy through mTOR, right? So what would be shutting off mTOR, AMPK, not MAPK, the opposite, right? AMPK, where AMPK is turning on autophagy. We're going to talk about this soon, and it's inhibiting mTOR, right? So this is interacting with um, raptor, and it's interacting with tuberin, and it is inhibiting raptor and promoting tuberin. It really does the opposite of what MAPK is doing. AMPK does. It's very catabolic, and the thing that activates it, yeah, exercise and AMP. You need to bind AMP to this thing so it can tell there's energy stress, and that permits activation by LKB1, liver kinase B1. Don't think of it as this little resident of your liver, right? This is all over the place. Now, it has effects in the liver, right? There are effects in the liver. It's like in liver, deletion of LKB1, you get some hyperglycemia, and so there are effects in the liver, but it's in skeletal muscle too, right? So its name, don't think that that's where it lives and it doesn't live anywhere else. A lot of these names aren't particularly descriptive of their residence or their function. But LKB1 in the presence of AMP, we're going to talk about this on Friday. It's going to be all AMPK on Friday. That's all we're going to talk about. And so AMP binds, permitting the activation of LKB1, and then once AMPK is active, we're inhibiting mTOR. And testosterone inhibits LKB1 also. So it inhibits the inhibitors of mTOR in addition to promoting. So testosterone does have a strong role in cell signaling cascades despite being a steroid hormone, despite having its classical, traditional, canonical signal be nucleic. Now, if you knock out LKB1, just because LKB1 is catabolic doesn't mean it's not critical to muscle homeostasis, right? That it's not critical to the metabolism and development of skeletal muscle. If you get rid of this thing, you're going to be in trouble. So just because something is catabolic doesn't mean it's horrible for your muscle, right? It's about balance. It's about homeostasis. It's about fitness. And if you want to grow, you lean in a direction, right? You don't execute everything that opposes that direction. You don't eliminate and execute opposition. What you do is you favor one direction or the other. That's how it works. And how we'll next week on Monday and Wednesday, we're going to be talking about the applications of mTOR and the interactions and some of these earlier phenomena we've talked about. Whether it's nutritional interventions, we'll sort of introduce, but size principle, stuff like that, and some of these biomechanical phenomena we're going to introduce. And in the way that we lean, we slant mTOR signaling toward atrophy or toward hypertrophy. Now, androgens are going to inhibit LKB1. Estrogen is going to stimulate it. Estrogen is weird. And Testosterone and estrogen both, you're going to see an increase in ion mobilization, calcium mobilization. And earlier this semester, I said, like, you get fat, you put on pounds, you just put on some. So I'm not trying to be insensitive by saying I'm literally put on fat, get heavier, and make sure that heaviness is adipose tissue, and your strength goes up. Your strength always goes up in these situations, and why does it go up? I mean, historically, there have been a number of explanations of like, oh, it's like wearing a weightlifting belt. You're tighter in there. Like, I don't know. I don't really buy it. But one explanation is increased calcium from just go on a low fat, low cholesterol, high fiber diet and lean out a little bit, and you're going to get lower testosterone. So that is an explanation why when you see the E Hondas of the world, when you see world's strongest man or world's strongest woman, I'm sure that must exist. I have not seen this, but really strong folks are not super lean, right? The stronger that people are, often the more heft is in that midriff. And there are multiple. Brian Shaw. I don't know who that is, but Brian Shaw with a bunch of exclamation points. I'm guessing Brian Shaw is a better description than E Honda in this, explanation for this image. And with estrogen, once people, lots of excess adipose for sure in the comment box. So with estrogen, somebody hits menopause and you expect muscle weakness, you expect muscle, degradation, atrophy, deterioration of human performance. And why? Why do you see that? Well, estrogen, the power gut in the comments, estrogen has anabolic effects. Estrogen has anabolic effects, and its primary effects are going to be steroidal. They're going to be their cholesterol hormones, right? They're steroid hormones, so they get into the cell and bind to their receptors and have genomic effects. That's the primary, but there are non-genomic effects as well. And you see the intracellular calcium concentration increasing, a rise in the intracellular calcium. And so it's not just an acute thing where you can see some sort of effect on performance acutely. And there's arguments in the literature that are very believable about where they are right in their cycle. You're going to see different amounts of estrogen, different amounts of testosterone, and that can acutely affect performance. It can chronically affect adaptation, but it can also acutely affect performance. And so some of these non-genomic effects, estrogen can deactivate, phosphorylate in an inhibitory way. Tuberin, tuberous sclerosis complex 2, right? Tuberin. And you know what that does? You know what tuberin does? Tuberin is impairing the activation of mTOR through hydrolyzing Rheb's GTP. GTP hydrolysis, where Rheb is hydrolyzing that GTP, and GDP bound Rheb. That's not activating. That's not promoting mTOR. You need GTP bound. So estrogen can inhibit tuberin, and it's mediated. So the estrogen induced activation of mTOR is mediated through tuberin and the small GTPase ROS homolog enriched in brain. That's Rheb, RHEB. That's Rheb. And so that little cascade, so that's estrogen's activation of mTOR. But as we saw earlier with LKB1, estrogen does have interactions with inhibitory stuff too. AMPK and LKB1, estrogen has these direct relationships in a positive way with those. And so estrogen is weird. It's not like testosterone, which is go, 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 grow, grow, grow. Estrogen has some pros and cons to human performance. We talked about that earlier in terms of things like elasticity, and yes, it's anabolic, but it's not quite reinforcing the tissues in a way that's going to prevent injury. And so estrogen is unique. Just like when I'm talking about Deptor, mTOR and Deptor, and like, yeah, it's an inhibitor. Deptor, this is an inhibitor for mTOR, but it's also an inhibitor for apoptosis, right? And so it has its own life that's sort of pro and con both. Estrogen is a little bit like that. Testosterone is just sort of it's con if you don't want to grow or if you have cancer or something. There's nothing but con for testosterone. But in terms of athleticism, in terms of muscle development, growth, increasing cross-sectional area, testosterone is all go, 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 grow, grow, grow. Now, insulin, IGF-1, we've spent a lot of time talking about these ones already. And, IGF has its binding proteins, 1, 2, 3, 4, 5, 6. And 98% of the IGF is going to be bound to those things. And technically, IGF can also bind to insulin receptors. Insulin-like growth factor, yeah, it can bind. So if you get rid of all those binding proteins, you're going to have a problem with some insulin signaling. But insulin, we're going to go through that PI3K signaling cascade. Let's go get down to PKB. And over here, this is going to be GLUT4 translocation. Over here, right, we're turning off glycogen synthase. This is glycogen synthase kinase. That's the enzyme that is inhibiting glycogen synthase. So once you get PKB or AKT activated, you're turning on glycogen synthase, and so you're assembling your glycogen, mobilizing GLUT4 to get sugar into the cell, activating mTOR, and you know what's in between there is tuberin and Rheb. FOXO, fork-head boxo. We're inhibiting protein degradation. We're inhibiting the atrophy, and this is good if you have you want AMPK activation of this if you have cancer, if you're concerned with that. But if it's growth, you want to stop that, and that's what PKB is doing. And remember, PKB is also shutting off PKA. Remember that interaction from earlier where protein kinase A needs to be activated by cyclic AMP. And so you have phosphodiesterase is being activated by PKB and wherever there's PI3K, and are activating phosphodiesterase, which is breaking down your cyclic AMP, so PKA doesn't get activated. So just remember that PKB, super anabolic. It's going to stop catabolism. It's going to turn on anabolism in a bunch of ways. Now, IGF, we're going to be doing both signaling cascades with IGF. This is MAPK over here. So both signaling cascades with IGF. Growth hormone, we talked a fair amount about in the last section. Growth hormone as the hypothalamic pituitary, from the anterior pituitary, we're actually getting our growth hormone. So the hypothalamic pituitary liver response. So from the liver, we're getting our IGF. So the hypothalamus, pituitary, liver, we get our IGF and our binding proteins, right? Binding protein 3 is going to be bound like 80% of the stuff. And IGF is what is doing most of this tissue interaction. Now, growth hormone is going to bind, and you do see, the JAK-STAT cascade. You see PKB, see MAPK. Growth hormone has a lot of signaling, but most of the tissue interaction, this is going to be an IGF effect. And when you start looking at, well, growth hormone, how we know the different roads now. We've been looking at this map, right? We know MAPK, right? We know PI3K, and we know some of these different roads. What road is growth hormone really taking? And it really turns out to be PI3K, PKB is the main one. If you take rapamycin, which just shuts off mTOR, right? That's you're on your way from Canada to Mexico. You're on your way, and you shut down I-5 at LA. I mean, it's sort of the last stop, or San Diego or whatever, Oceanside or something, whatever. You're sort of at like the last stop before you get down to LA, the last major hub. It's all the upstream stuff doesn't really work. So anything north of San Diego gets shut down if you introduce rapamycin. All these upstream variables, upstream promoters and inhibitors. But what's more interesting is if you introduce an MAPK inhibitor, growth hormone still works, still grow, still getting muscle interaction, still getting hypertrophy with an MAPK inhibitor. But if you introduce warp-menin, remember, warp-menin is going to inhibit PI3K. That's a PI3K inhibitor. Now you're shutting down the effective growth hormone on tissue regeneration and hypertrophy. So growth hormone, it takes the highway. Growth hormone, yeah, it sends a little bit of some passengers down some parallel roads. Sure, yeah, it does. But growth hormone is really putting a lot of gridlock on I5. Growth hormone, its major effect is going to be going through uh, PI3K, phosphatidylinositol 3 kinase. Thyroid hormones. Now, we talked about this. They're a little bit weird. I don't really care about the intracellular stuff. Yes, they get inside of the cell. They sort of mimic what a steroid hormone would do to a degree, but also they have a PKB dependent effect. So that's that same highway, PI3K, PKB, that same highway. You're seeing thyroid hormones doing that as well. So endocrine system in review as it interacts with mTOR. We talked about all of the axes before, and all the hypothalamic activity and what the pituitary does and stuff pre-mTOR lectures. But now those hormones we introduced, here's how they interact with mTOR. Here's how they interact with muscle. Right? This is a muscle physiology class. And so how do these hormones affect muscle? Insulin, bunch of PI3K, right? That road, I5, PI3K, PKB, down through mTOR and straight to the ribosome, and let's link together some aminos. Thyroid, same thing. Thyroid hormone, same thing. Human growth factor, IGF, mostly PI3K, right? Yeah, we got some other, there's some additional stuff here. But the primary signaling for these is going to be PI3K. Testosterone, we have calcium dependent MAPK, mitogen activated protein kinase, RSK, ERK, that signaling cascade. We have that being activated, and an increase in IGF signaling. So we're getting more of this, more IGF interactions, and then inhibition of LKB1. And we're going to talk more about this as we get into AMPK on Friday. And estrogen, a little bit weird, right? Estrogen is sort of your untrustworthy friend who sort of comes through in the end. And you can tell because, do you remember in The Office when Dwight left and all the plants died? It's like, what nobody, his work was sort of invisible. For those who watched the real Office, the good one, the one that actually was funny, the American version, uh when Dwight was fired, or what, I think he like shot a gun in the office or something, was let go for whatever reason, he's gone, and all the plants die. Get rid of estrogen and your tissue, it's hard to retain that stuff. If you ask your moms if they are going through menopause or have gone through menopause, just like physical exertion and vigor and maintenance of lean body mass, it's tough. So estrogen is the good guy, estrogen is the good gal, right? But it's a little bit tricky because you're seeing some promotion of LKB1 and AMPK, but there's also good effects to it as well. So the endocrine system sort of in review. Yeah, all these growth factors and sort of lumping in everything else here, the PI3K, PKB signaling. And the goal is to get Rheb to activate mTOR. The goal of all these hormonal things is to get Rheb to activate mTOR. Whether it's growth hormone, testosterone secondary effects, estrogen secondary effects, and thyroid hormones and insulin-like growth factor, and a lot of these chemicals that we talked about, IL-2, IL-15, prostaglandin, whatever. The goal for a lot of these things, maybe not as much prostaglandins as we're looking at MAPK, but let's make Rheb activate mTOR. That's really the goal of this stuff. And there's a location that matters. You can't do it if they're not at the work site. You can't get mTOR to work if it's just hanging out leisurely in the cell. You need to send mTOR to the lysosome. This is its office. This is where it works, right? Get mTOR to the lysosomal surface and it's ready to work. Send it to the office. Now, this stuff, all these growth factors, what we've talked about so far doesn't do that. What we've talked about so far activates mTOR if it's at work. If it's at work. Now, the lysosome, the reason you keep seeing it and the reason I keep bringing it up and you keep seeing it in these diagrams is it actually is important. The lysosome itself is not, it's like 2% of the cell. It's not huge. It doesn't take up that much. It is, you're degrading a bunch of things. It has, I don't know, 60 plus enzymes in there. It's just breaking stuff down. It has these degrading enzymes in it. It's breaking stuff down. Think of a lysosome as a recycling plant, right? You recycle your metals and a little bit of plastic and some paper and whatever. You make new stuff out of it. You make new stuff out of the old stuff. And so lysosomes are melting down your metal. They're melting down metal, and you can make new things out of that old metal. So mTOR has to actually go there. Now, if you think about like mitochondria by comparison, mitochondria is super, super important. I don't know, 10, 12, 15% of your cell volume. Lysosomes are small. I was going to say no less important than the mitochondria. That's not quite true, but they are critically important. They function as these amino acid storage sites and a sensor for amino acid content. If you think of them as these recycling plants, if you think of them in that way, they contain the raw materials that you're going to need. Now, what is mTOR doing? Well, it's translating a bunch of proteins. It is not mTOR itself, but mTOR is the sort of floor manager who's responsible for dictating translation. And yeah, you need the raw materials. And so that's why the lysosome is so important. Is it is mTOR's work site. You know, it's the farm, right? It's not druids hanging around at Stonehenge where it's just this feels like a nice spot. This is the farmers working with in it's Dwight in the beet field, right? It's the Schrutes wandering around watering the beet field. You have to be in the farm to get the crops, right? It's important to be at the lysosome. And so now as we move in to the amino acids, when we move into protein, this is about transporting mTOR. It's the taxi that delivers mTOR, the complex, to the worksite. So what we know so far, mTOR is activated by a lot of things. mTOR is turned on by a lot of things, right? We have the immune system, chemicals turning it on, interleukins and interferon gamma, the Wnt proteins, tumor necrosis factor. Myostatin would be a negative regulator, but you could say follistatin here as a promoter, and mostly PI3K with some residual traffic traveling the MAPK highway. But that big 6-lane highway, this is PI3K and just gridlock. I mean, it is just kind of bumper to bumper on PI3K and some spillover on MAPK. Mechanical tension works in a lot of ways, right? There's MAPK, there's PI3K, there's DGK, phosphatidic acid DGK, stretch activated channels. There's probably more stuff. Mechanical tension works in a lot of ways. Some of it is going to be through that canonical pathway. Endocrine system, mostly it's going to be a bunch of PI3K. Largely, yes, there's other stuff too, but PI3K, PKB, that cascade. But you have to actually get mTOR to the worksite. You have to have the bus or the taxi, the broomstick, if it's like Harry Potter, whatever it is. You have to get mTOR to the worksite, and that's where nutrition comes in. It's not necessarily where carbohydrates really come in, but it's especially where amino acids come in. So we have whatever you're putting in your mouth, right? You have to have fitness, right? You have to have a nutrient sensitivity because scarcity has been the great history of human experience. Nutrient scarcity, whether it's bad hunts or famines, and there's a famished history in our species, and so we have to be able to detect nutrient scarcity. And in the presence of ample nutrients, we respond, right? Okay, now we have the ingredients to make our proteins, but in the presence of scarcity, we have to have an internal appetite for that. Remember the way I talked about the soft shell crabs feelers, right? The important bits are inside, but they reach out their feelers a little bit to see if, okay, now's the time we can migrate. Now's the time we can search for food. Now's like a time we can play on the sand or something. Or if you if you owned them as pets, you had little tiny castles, they could, do laps around the castle. And with your appetite, it's the same thing. It gives you this little signal to say, is food available? My stomach growled and I'm a little bit empty feeling. Is food available? If you put food in your mouth, you know that food is available. And so we have to be able to detect nutrient availability or scarcity. And amino acids, these proteins, there's a bunch of proteins that are going to do that in the body, and they're going to respond to amino acids. And that's the RAG GTPases are really what are going to do that. So over here, you see RAG GTPases. This is how you're sending mTOR. This is the taxi. This is how you're getting mTOR to the worksite. Regulator, you're regulating this stuff. I mean, I'm going to talk ridiculously little about that, but the RAG GTPases, you should know that. Now, VPS 34, this is a type of PI3K. Wortmann is going to inhibit it, right? So VPS 34 down here, that's a type of PI3K. But the main effect of these amino acids, right, it's a RAG GTPases. And this is just saying, when you see regulator all over the place, like, what the hell is regulator? Here's, as far as I'm aware, is David Sabatini. This is a huge researcher in the field. The coinage of regulator. We refer to this complex as regulator, and regulatory roles. You don't really need to know about that, but you do need to know about RAGs. And earlier I mentioned that lysosomes, and you're going to see a lot of David Sabatini's work. I mean, he really is one of these primary foundational researchers in this field who's done a lot of the work that has advanced our understanding of mTOR. So lysosomes can be loaded with amino acids sufficient for the recruitment of mTOR, and intralysosomal amino acid levels matter. So cytosolic amino acid levels matter, but also intralysosomal amino acid levels matter. And so here's how it works, just this sort of brief glimpse at what you have is RAG, especially over down here, this GTP here. This actually binds to Raptor. So again, I said Raptor has a lot of interactions. Raptor is so critical. It's this point of interaction for so many things. And RAG, that's going to bind to Raptor. And then we mobilize mTOR to the lysosomal lumen, to the surface of the lysosome. And here we have Rheb, right? We have Rheb here, and we're facilitating interaction with Rheb. So this is the taxi, right? This is the commuter that's going to deliver mTOR. It's going to bind to Raptor and deliver the mTOR complex. You see the complex here, MLST8, right? That's Tom Cruise. That's Lestat. That's Gable, G protein beta subunit like. That's G beta L. And there's Raptor and the mTOR complex. And so RAGs, RAG, GTP, this is going to bind to Raptor, and that's what's going to mobilize it. That's what's going to move it and get it to interact with Rheb. Now you have to turn on Rheb, right? This doesn't turn on Rheb. You need some sort of growth factor to turn on Rheb, and that's what we've been going over as we went through the endocrine system, as we went through mechanical loading, as we went through the immune system, the activation of mTOR. But this is delivering it to the actual worksite. So what amino acids are we talking about, right? So you have to sense amino acids and get them into the cell. Well, how are we sensing them, and what amino acids are we actually sensing? Here's the 20. Here's like the 20 normal amino acids. And there's a few of them that are more important than the others for mTOR signaling. And when you look at, so this is P70SSK, this is a phosphorylation site. mTOR phosphorylates a particular site here. And what does that phosphorylation look like in the presence of specific amino acids? This is more of David Sabatini's work. And no amino acids, you would expect that. Like, well, okay, so you're not getting mTOR mobilized. All of them, right? Just introduce all of the amino acids, and okay, you get like 100% of mTOR activation. And just leucine and arginine, that's not bad, right? That is not bad if it's just leucine and just arginine. If it is uh, just leucine and just lysine, that's not very good, right? There's a little bit, yeah, just a little bit there. Just arginine and just lysine, there's like nothing going on, right? So leucine is sort of the most important one for activation, but just leucine by itself, you're not really going to get anything happening. That trinity, right? Leucine, lysine, arginine. That's what I've been talking about this semester is leucine, lysine, arginine. You get about 90% of the activation. 90% of the phosphorylation, the activation of this. Now, if you want to introduce a fourth player, right? If you want your trinity to be a quatrity or whatever, a quartet, then methionine, that one, the mTOR complex. I'll show you what that means a little later. Less important, but that would be the like reserve champion of the fourth place. You didn't quite get bronze, but your honorable mention, methionine, that would be up here. So that one is our honorable mention, but leucine, probably the most important. Arginine, second most important. Lysine, third most important. And I'll talk about how the sensing of those things work, but the muscle, it doesn't really seem to just sense everything. Yeah, it will probably, I don't want to take that sentence back in 5 years when additional protein sensors have been identified. I don't want to have to take that back. But what it appears to be is there's some proteins are really sensed and others, I don't know, other proteins can sort of fit the leucine pocket if you really, cram them in there. But when I teach stats, when I teach epidemiology, I talk about inferential statistics and making inferences based on a sample. You sample from the population. You don't just evaluate every single person in the world whenever you have a research question. You randomize like 50 people into your study and then make some inferences. You analyze it and make inferences about 1 billion people out of 50. And that seems to be how the cell sort of behaves, where there's some amino acids, my complete protein, fine, that's wonderful. Complete protein is wonderful. But I'll bring this point up again, but evolution didn't seem to have the foresight to uh, expect chemistry labs to be isolating amino acids, where you can just take a leucine by itself and let me just take a leucine and arginine. Evolution didn't really have the foresight to anticipate, chemistry. And and so it just is like, all right, let me identify a few amino acids. And if those are present, you probably ate a bite of meat and let me just behave accordingly. So it is back in the assembly of our genomes, that was probably wise, very efficient to just identify a few and say, all right, they're probably all there. I don't need an entire head count. I don't need to keep monitoring. I'm seeing you got some leucine, you got some lysine, you got some arginine. I mean, you got some amino acids in here. I'm guessing there's a bunch more where this came from. Not always how it works in the supplement days. And so this is one domain in which exploitation of the system is a possibility. Now, how are we sensing these things? What protein complexes exist in sensing? Well, there's probably, in terms of mTOR's movement to the lysosome, there's at least 30 proteins involved and just moving mTOR along. But the sensation, earlier I mentioned intralysosomal amino acid sensation. This thing, which looks like an astronomer's star identification or like a bad license plate or so. This is a transmembrane protein, SLC38A9, horrible name. This is a transmembrane protein. You know what those are. They cross the membrane. And so we're at the lysosome, the lysosomal membrane, like integrins, transmembrane protein, but they're up at the actual, cell surface, sarcolemma, they're reaching across. And for the lysosomal membrane, this is what's sensing intralysosomal arginine. So how much arginine exists inside of the recycling plant? That's what's sensing that. And what's important about lysine, lysine seems, which is why it's the least important of the trio, it just seems to be arginine's sidekick for its major roles. Yes, research continues, and lysine's involvement, I'm sure, has a bigger role. It's not just this little tiny minor character, but sort of compared to arginine, it is. And so lysine, what it seems to do is promote the interaction between this protein and this regulator. Promote that interaction. If you are deficient, if you deprive a cell of lysine, you see a poorer interaction here, sort of the mobilization of mTOR with this particular mechanism. And now, if you uh, deprive a cell of arginine, that has a much larger inhibitory effect than if you deprive lysine. But that's where lysine comes in too. But that's not where arginine stops. Arginine also is detected by castor, CASTOR, C for cellular, A for arginine, S for sensor, and then TOR, obviously. So the cellular arginine sensor for TOR. And this is out in the cytoplasm. And so you are sensing arginine out there too, because if you just inhibit this thing, you can still sense whether you have had some arginine, and whether the supplement has entered your mouth and stomach. You can still sense it. Yeah, it's burdened by your inhibition, but there's still sensation. And so this is probably the best diagram that I've ever seen that shows the effect of amino acids on the mobilization of mTOR. Now, you know the activation of mTOR, that's Rheb, right? That's the tuberous sclerosis complex, TSC2, tuberin. Tuberin is facilitating the hydrolysis of the GTP, so that if you do that, Rheb isn't going to activate mTOR, right? If you do that, you like shut off the internet on campus or something. So mTOR takes its taxi over to the campus, but there's no internet. The buildings are locked and whatever. That's what tuberin is doing. It's preventing mTOR from working while it's at work. So you need both of these pathways to be active. You need amino acids available, and you need some sort of either carbohydrate ingestion to get insulin, or you need growth factors and chemical responses from use, mechanical or tissue damage, something like that. You need both of these doing their work. Probably the best diagram. But the funny, ridiculous, obnoxious part here is that it's a quadruple negative. So Gator 1, this is the main spot of protein sensing out here in the cytoplasm. The single primary spot of protein sensing, if you're going to identify one, is Gator 1. And Gator 1 is inhibiting this. So Gator 1 is inhibiting RAG's mobilization of mTOR. Now, and Kickstore, I'm never going to ask about this, but this recruits Gator 1 to the lysosome, just so you know what it is when you see it. This VATPase, vacuolar ATPase, you know what an ATPase is, but this is you are regulating your pH, right? You're pumping protons around, and not going to ask about it, but just so you see, so you recognize everything that's on here. And again, regulatory, not going to talk about this regulatory thing, but Gator 1 is halting, it's inhibiting, it's a roadblock to the taxiing of mTOR to the workspace. Gator 2 is inhibiting Gator 1. Okay, Cestrin 2 is binding to and inhibiting Gator 2. And Castor 1, we talked about Castor, that's a cellular arginine sensor of TOR or whatever. And so that one is also inhibiting Gator 2. And leucine if it binds testosterone, is preventing this interaction. It promotes the dissociation if you bind leucine testosterone. So what we have, again, quadruple negative. So if you thought tuberin is inhibiting Rheb, um, and so like PKB shuts off tuberin, which is shutting off Rheb, and Rheb activates mTOR. A double negative is obnoxious and confusing. I mean, the amino acid stuff. This is like arginine inhibits Castor's interaction with Gator, which shuts off Gator 2's inhibition of Gator 1. And Gator 1 would have been inhibiting the RAG GTPases. And so that promotes it. So it's a negative, 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 negative is a positive. Four wrongs in this case make a right. If right means translation, if right means hypertrophy. Now with leucine, introduce a bunch of leucine, right? It binds to cestrin 2, and normally cestrin 2 is going to be bound to Gator 2, shutting it off. But if you put a bunch of leucine in your mouth and swallow it and get it into your cells, you can inhibit that. You can inhibit that response. Now, people take leucine very chronically today. That there's really high doses in the bodybuilding community of just leucine, leucine, leucine all the time. Let me put in 50 grams of leucine every day, and because it's the most anabolic of all of the amino acids. But when you do something like that, when you're sort of ODing on leucine, it's likely that just cestrin 2 expression is going to increase, because remember, metabolic homeostasis, metabolic balance, grow all the time, your body's going to build up a couple of safety nets. And one such net could be increased cestrin 2 expression. And I don't know what that would look like, or the time course of manifestation, or the magnitude of it. But realizing excesses of supplements may bear some consequence in genetic expression. Possibility. That's a possibility. So that's how arginine and leucine are working. Now, arginine is also working in the lysosome, but cytosolic sensation of amino acids, castor and cestrin 2, and that works through the Gator complexes, which Gator 1 inhibits RAG, the taxi mobilizing mTOR, to uh, the lysosome, right, to where it's actually going to do its work. And you don't need to know this, but there are transporters to get these things around. There are more and more and more proteins become involved. Regulatory proteins get drawn into this, and so I mean, there's just so many checks for the balance. These checks and balances to keep metabolism, to keep protein turnover balanced. So many checks, so many proteins sensing and regulating all of this stuff. And just as a nice little recap of all of this, of Sabatini, a little bit old. This is a decade old, but mentioning that there's a couple of things that have to happen, right? So we're going to have to go through the mostly PI3K or MAPK, but mostly PI3K signal to get mTOR to be activated, to get Rheb to activate mTOR, to get mTOR turned on. We have to do that signaling cascade, but we also need amino acid availability to mobilize it to that spot. And so this is just a look at upstream stuff. And so here we see tuberin, AMPK is turning it on, MAPK is turning it off, PKB is turning it off. So it kind of doesn't really matter where you start. This is epidermal growth factor, but any growth factor, who cares? Growth factors up here. I mean, it is IGF and whatever. And you have interleukins that are activating MAPK. There's growth factors and chemicals and whatever, hormones, and insulin going through PI3K. We're activating Rheb, but we need to mobilize it, right? We need to mobilize it to get there. But all this stuff should really start to uh, it makes sense when you see MLST8 or Gable, same thing. That's the same thing. And Raptor over here, all of these different phosphorylation sites of Raptor. Remember, Raptor can be phosphorylated in a promotional or inhibitory way in multiple different sites downstream from mTOR. Raptor is recruiting as downstream targets, and mTOR is phosphorylating them, provided it is at its office. It is at the work site. And there's the RAGs, right? So amino acid RAG GTPases mobilizing mTOR to the work site so that you can function. Pras40, you saw this. Deptor isn't on this one, but Pras40 is a negative regulator of mTOR. Interacts with FKBP12, interacts with MLST8. It inhibits mTOR's kinase activity. We'll call that one a core protein. Fine. It's not as interesting as Deptor, but Pras40, that's another negative regulator. But everything on here you recognize. Everything on there is recognizable. Now, this counter, this opposing signaling from LKB1, get low on nutrients, and let's activate AMPK, this opposing position of here's PI3K. Now, this is a little bit backward. So PI3K, remember, is converting PIP2 to PIP3, phosphatidylinositol bisphosphate to triphosphate. P10, phosphatase intensive homolog, this thing is converting PIP3 back to PIP2. So it's not really, it's not inhibiting PI3K. It's just the opposite enzyme. This is a kinase. This is a phosphatase. They're doing the opposite thing. But the arrow is fine. Whatever. It's opposing PI3K's activation of PKB by way of PDK. And PKB is inhibiting tuberin. Tuberin is inhibiting Rheb. Rheb is activating mTOR. So Rheb is the activator of mTOR, direct activator of mTOR. You know the downstream targets. But AMPK over here, I mentioned mitochondrial biogenesis before, and mitophagy, but we're all this autophagy, autophagy. The phagocytic behavior of oneself, right? Autophagy. So LKB1 is this stimulator of AMPK, but you need stress. You need physical stress, not like emotional stress, but you need a bunch of AMP to bind to AMPK, adenosine monophosphate activated protein kinase, so that LKB1 is able to activate it and promote tuberin and inhibit Raptor. So LKB1 does tons of stuff. LKB1, tons of functions. And earlier I said it's even critical to muscle metabolism, where if you get rid of this thing, you have problems. It's not like, well, I blocked it. It's like, oh, myostatin knockout, I'm huge. LKB1 is not quite like that. The only interaction I care about, though, is AMPK. The LKB1 is the promoter, this primary upstream promoter of AMPK. And it is a primary regulator of muscle fitness, of muscle physiology, metabolism, homeostasis. This should also make a bunch of sense, right? You should start recognizing these clear maps. There's a couple things on here that you're not going to P38 and PRAK, that P is just P38, regulated, activated protein kinase. I don't care. Who cares? Who cares about that one? Energetic stress, you are exercising, right? You are on a jog, you're on your recumbent bike, you are doing whatever it is that your exercise, your sport calls for. You're on the soccer field, energetic stress. Or just low carbohydrates and low sustenance intake. That's also energetic stress. So especially when you combine those two, when you're on a diet and exercising, if you're exercising within the presence of nutritional scarcity, you're going to get even more AMPK being activated, and then you'll see promotion of tuberin and inhibition of Raptor. And so this is an inhibition of mTOR. But growth factors, you see PI3K up here. remember downstream from mTOR complex 2, PKB is one of its sites. This looks like a Thanksgiving turkey or something. But downstream from PKB, the inhibition, the phosphorylation sites of mTOR. Raptor recruits them. Amino acids, RAGs, right, via RAGs are going to mobilize mTOR to the worksite. Raptor recruits its downstream targets, and now mTOR phosphorylates them, and away you grow, right? You just start growing at that point, because you have mTOR activated by its upstream targets. Rheb has turned on mTOR. You have mobilized mTOR using the RAG taxi to the lysosome. And then while you are there, some upstream thing turned it on, and now mTOR is phosphorylating its downstream targets. And this is just where I said, like, okay, there's more to it than with, like, when you look at reactive oxygen species are playing a role here, mediating the effects of leucine on translation. Okay, so reactive oxygen species do rear their confusing head every once in a while. And there's a lot of anabolic stuff that they play, but pros and cons, right? They have pros and cons. Now, again, as I said, last lecture or lecture before or something, I wouldn't go take the bottle of vitamin C before you go start exercising. Not just because of mTOR, but I mean, look at things like adiponectin release, and there's all of these interactions that reactive oxygen species play, that you want to have them play out, right? You don't want to bench them. You want to leave those on the field so that they will collaborate with the players who are on the field. Okay, so for nutrition, our our kind of glimpse over today's shoulder, fats, right? Okay, well, lipids, we have prostaglandins and phosphatidic acid, right? These are lipids. And so this is, diacylglycerol kinase to convert a diacylglycerol to phosphatidic acid, and prostaglandins come from arachidonic acid. These are lipids, and then androgens and estrogens. These have secondary effects on mTOR signaling. So fats, if you're going to be putting food in your mouth, I wouldn't go, not necessarily for the reason of like prostaglandins, but for the reason of androgen secondary effects. I would not be denying fats from the diet, these low fat diets in terms of human performance. We're going to talk a little bit more about this when we get into the mechanisms or the applications, strategies for applying this information. And carbohydrates. Okay, we're going to talk on Friday about AMPK. We'll have a conversation about Friday about AMPK. We'll talk a little bit more about its activation and its inhibition, but glycogen can bind to the beta subunits, and there's going to be an inhibitory effect on AMPK. And carbohydrates are also going to elicit, I mean, leucine will too, some amino acids more than others. If you eat protein, or you're going to get a insulin response, not a very big one with amino acids. You'll get a bigger one with just go eat some white bread or something. But carbohydrate ingestion, you're going to get an insulin response, and you're going to stimulate PI3K. So carbohydrates, anabolic. Proteins that we just finished talking about. Proteins are very anabolic from the perspective, especially, of their ability to mobilize the mTOR complex via RAGs, so RAG GTPases, to the lysosome, where it's going to do all of its work, where it can interact, where it's free to interact with Rheb, provided you have other stuff to activate it. And some amino acids are more important than others, right? You have leucine, you have lysine, you have arginine. Those are the three most important ones. But remember, we have SAMTOR, it's a methionine. This is the fourth place one that receives the piece of paper that says honorable mention or whatever. So combining all of this stuff as we begin to summarize the day, and it looks like we might actually finish class on time, as we summarize all of this information about the activators of mTOR. I'm just going to read these. I mean, I just wrote them, but I wrote them in such a way that I think it should illustrate the point, where if you're using a cell, if you're using muscle cells, Henneman's size principle, you have to consider Henneman's size principle. You may need to repair the cell that you're using. Now, will you get responses in adjacent cells, neighboring cells? Will you get an mTOR response? Maybe, yeah, the whole community may benefit when one household really invests in reinforcing their remodeling their house. Maybe the whole street is going to increase their home values without doing anything. A little bit similar in mTOR. Sure, we cells are not isolationists, right? We have these transmembrane proteins like integrins and cadherins, and we're familiar with what the neighbors are doing. But if the neighbors are the ones, if one cell is electrically activated, it was their actinomyosin that did the labor. If one cell is electrically activated, it was their energy cycles that consumed all of that sustenance, right? It was their metabolism and their um, mechanical productions of loads, and it was their responsibility. They're reinforced by it. There may be a little spillover, sure, but the cells that are working, those are the ones that are reaping most of the rewards, the bulk of the rewards. So when a cell is damaged, you get an inflammatory response. It's a bunch of immune chemicals, and cytokines, chemokines, immune chemicals, these prostaglandins, and these are a marker for how much damage there is. Right, the more damage, you do 20 sets of squats or one set of squats. You do your Alfredson protocol. Do you do 100 negatives, those eccentric drops? Do you do 100 of them, or you do 10? Right, the amount of damage is going to dictate the amount of that inflammatory response, the amount of architectural disruption, right? You'll be more sore, you'll have more chemotaxis if you do more damage, if you disrupt more tissue. And the response of that is even more signaling, even more cell signaling, more PKB, more MAPK. Yeah, more pain, more muscle soreness, but also more signaling. And the mechanical loads are being transmitted to say, here's what the cell is doing now. And this telegraphs to the interior of the cell what the cell is likely going to need in the future. Here's what's coming out of my wallet now. Here's what I'm going to need in payback later. So you have mechanical signaling saying, here's what I'm doing now. You have the immune system and hormones saying, here's what we just did. That's need. That informs the cell that need is in play, that you have to respond with protein translation to reinforce those cells. Now, nutrition, so need is one input of mTOR. Nutrition is another input, where you don't want to grow if you don't have the resources to do so. You don't want to overspend out of your bank account if you don't have the food in your mouth, the money coming in every month, right? So you have to have adequate carbs, fats, and proteins. You have to present your cells with adequate nutrition to stimulate its growth, to say, hey, look, I'm giving you the resources so that you can spend. It's sort of a tax and spend. The government can't spend money, whatever, just like borrows from China or whatever, but the government's supposed to tax a bunch of money and then spend that money to repair roads and repair bridges and build new whatever schools and stuff. So nutrition, you need the appropriate nutrition, the assumption of nutrition, and that's where like insulin and stuff comes in, where you can provide the calories, and the body's like, okay, I see calories are coming in. I now have the assumption that I'm going to have ample finances to support this project, to support this architectural development I'm building, but also the assumption of nutrition coming from like insulin again. That if going back to this idea that evolution couldn't anticipate the creation of a hypodermic needle. So you can just give your body the assumption of nutrition when it doesn't actually have it, right? You can give yourself leucine, lysine, arginine, and an insulin injection. And like, wow, man, I'm really growing, but you don't really have nutrition on board. So you can fool your body. Now, the combination of activators of need and nutrition, and this is really what's doing it. So we have if you're just eating a really well balanced diet, tons of protein, tons of carbs. Over here with the protein, you're mobilizing mTOR to the lysosome with all these rags with the amino acids, and with all those carbs, you're getting insulin and activating mTOR with Rheb. A really well balanced, just eat a ton of food and don't miss out any key nutrients. You can get mTOR to be activated in doing so. But you can also get mTOR to be activated by just introduce a bunch of nutrients, and then you don't need insulin, just go exercise. Activate the PI3K PKB route, that road, activate I5 by some other means. You can do it that way. So a couple different mechanisms, a couple different ways of activating mTOR, but you can't just do one. You can't just do one of these. So, I just ate a ton of protein and I'm not like exercising and disrupting tissue, and I didn't eat a bunch of carbs, and I didn't. Well, okay, you mobilize mTOR and it gets there and it's like stood up. It's like, well, there's nothing here. Where's my Rheb? I have no, workplace resources. I'm unable to do my kinasing, my phosphorylating. Or it's just like, well, I ate a ton of carbs. I have this carb heavy diet, and I did a little bit of exercise or whatever, and I have some MAPK going. I got some IGF, but like where mTOR is nowhere to be found, right? You haven't translocated it to the lysosomal surface. So you need both of these, and it's a little bit like this is the Nuclear Surety Tamper Control and Detection Program from 2013, and by order of the Secretary of the Air Force, Section 1. Item 1.1, the very first thing this says, this little 8-page document that says, all right, here are the policies from official statements from the Air Force. Section 1.1, Tamper Control Program, the two-person concept, meaning if we're going to have some nuclear activity, you can't have one person doing it. That one person could be biased. That one person could be pissed on a day. That one person could be drunk. That one person could have a whatever it is, they could have accidentally done something. There could be a little mishap. There could be any number of things. You have to have a two-person system. mTOR works like that two-person system, where what you need, I think Gringotts, right, in Harry Potter, like the Goblin Bank, I think they have like 2 keys or something. The actual person and the Goblin key. This is standard, and it comes from your body. Right? This is a biological phenomenon to prevent, if it's a one-person system, well, now you have some, if you mutate rags, they've done this. There are experiments where they've done this, not in like people, but you get this mutation of the rag proteins, and you get runaway metabolism, and life doesn't last very long. So you need this two-person system. That's how mTOR works. There has to be proteins available because those are the ingredients. You are linking together a bunch of amino acids. You're making proteins. You need proteins available. That can come from the lysosome or what you're eating. And then you also need growth factors to do the on switch. So that is everything for today. What questions do we have?
Real quick. So going off of leucine, lysine, arginine, rag, and the carbs, and basically the last slide you just said, I think it was a year ago, I looked up a video out of curiosity of some bodybuilder who's on keto. Now, he said to get what he does to maintain size and to get bigger is reps of 30 with like stupid lightweight, like something like my little sister could do. So I guess what I'm trying to ask is, based on what you just told us about, with all this stuff and Rheb and insulin, is he just saying this to appease to people and based off of like the nutrition of keto? Like, is he just promoting keto in a sense with some like false placebos? Oh yeah, if you do a lot of reps, it'll work. Do the fat burning, blah, 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 like ketosis and all that. Or is there any scientific evidence that this man is possibly right? Because I understand that eating the protein, you will maintain your mass. I mean, as you said, you have leucine, lysine, arginine, but there's not that main, there's no insulin, there's no carbs. And I guess what I'm trying to say is, yeah, is this basically just a fitness advertisement, if that makes any sense,
It's
this whole keto movement?
closer, there are levels of truth here, and I would say it's probably now, without listening to this person's sentences myself, and
Mhm.
um, my hunch is it's closer to an advertisement than it is to, a sound physiological discourse or whatever. But you can totally grow on a ketogenic diet. It's just you have a very slight barrier of insulin with no sort of anabolic surges of insulin, where you get this insulin, but you can activate it through other means. And so carbohydrates. If you want to just grow to the best of your potential, you want to be huge, you want to be Ronnie Coleman, right? Okay, I don't eliminate carbs. It's not going to be as effective, but you can activate those pathways in other ways. Now we'll talk about if he's lifting light weights and doing 30 reps or something like that, but he's occluding blood flow. If he's like wrapping bands around his arms and then doing curls and occluding blood flow, I'll go over that, where you can actually see some mTOR stimulation that way. And on a lighter weight, you can trigger mTOR. That'll be in our application section. I'll talk about that. But you should activate the high threat if you're like young. I mean, if you have osteoporosis and you're at risk of cardiovascular adverse events, maybe don't go do a bunch of high threshold activity. It's certainly at least on like day one. But although if you are at risk of adverse cardiovascular events, I wouldn't do a bunch of occluded blood flow, ischemia, reperfusion um, exercises either. But there's truth to the philosophy. Whether there's truth to his sentences, I would have to hear the sentences. There's truth within the philosophy, and ketogenic diets present a barrier. It's not insurmountable, though. Just because you're not getting a big insulin response, and also when we get to AMPK, the glycogen can bind to the subunits, the beta subunits, and um, so just because there's a couple of hurdles to leap doesn't mean that keto is a difficult nutritional system for growth. You just have to be more conscientious or something. You have to be a little bit, you have to dial in your exercise a little bit better. And especially, I mean, if you're just doing like steroids at the same time, who cares? Like, everything works. Yeah, whatever, it'll be fine. You're getting your PI3K and MAPK and straight steroidal effects of testosterone. Eat nothing. If you, I mean, it's just like so it's in certain bodybuilding communities, if it's not um, if androgens are on board, it messes up your. It sort of messes up some of my sentences, but if androgens aren't on board, if you're not doing exogenous steroids, then you just have to be a little bit programmatically careful. Yeah, you have to manage it a little bit better and make sure that you get PI3K, PKB signaling from other sources rather than big insulin surges.
Awesome, thank you.
Anyone else? I have to go in just a second because somebody's coming to look at my carbon monoxide um, poisoning household. But one more question, if anyone's got it. Okay, I'm going to go address the air in my house, and I'll see everybody on Friday for AMPK. We'll go through AMPK, and then on Monday, we're going to do applications of mTOR. Might last to Wednesday, I'm not sure. And then we'll start rehashing the information that we've gone through a little bit to make sure here are the important points. Here's what I want you to remember. All right. I'll see you guys.